Abnormal DNA methylation and histone modification patterns in the APP gene that contribute to the pathogenesis of Alzheimer's disease. Amyloid precursor protein, APP, is a protein that is highly expressed in the brain and is connected to Alzheimer's research because of its involvement in neuronal growth and repair, synapse formation, and cell signaling. Recent research has shown that abnormal DNA methylation and histone modification patterns in the APP gene lead to overexpression, which could eventually lead to neurodegeneration. The neurodegeneration is observed by comparing the brain tissue samples of patients without Alzheimer's disease, the control group, and patients with Alzheimer's disease. The main findings from the various research articles that were used to show that there is a notable hypomethylation in the promoter region of the APP gene, leading to abnormal gene expression compared to the control group. Since there is no cure for Alzheimer's disease yet, understanding the epigenetic changes allow one to see the molecular mechanisms and create new treatments with this. The hypothesis is there are epigenetic profile differences in the APP gene between Alzheimer's disease patients and healthy patients that can be seen through the analysis of epigenetic data.
Alzheimer's is a neurological disorder that affects memory, behavior, and thinking. Many symptoms of this disorder can get severe enough to interfere with daily life, as it decreases cognitive function due to an increase in amyloid and tau proteins in the brain. This disorder affects 7 million Americans, and that number is projected to increase to nearly 13 million by 2050. The share of Americans over 65 years old is increasing, so more people will develop Alzheimer's in the future. The signs of this disorder tend to appear in individuals over the age of 65. Some common symptoms of this disorder include a memory loss, inability to understand language, confusion, a decrease in physical coordination, and mood or personality alterations. Currently, there is no cure, but there are medications available to aid in managing and inhibiting the progression of this disorder. Research has found that amyloid precursor protein, APP, is a protein that is concentrated in the synapses of neurons and is important to synapse formation, growth and repair of neurons, neural plasticity, and cell signaling. DNA methylation and histone modification are crucial for regulating gene expression, DNA replication, and repair. We examined recent research indicating that abnormal DNA methylation and histone modification of the APP gene lead to overexpression, which causes degeneration of the brain. By identifying abnormal methylation and modification in the APP gene, this can allow for more medications and treatments to be developed for this disease.
A comparative analysis of epigenetic data from published research articles were used to identify differences in DNA methylation and histone modification patterns. The samples analyzed from the brain region were the cerebellum, anterior parietal lobe, and inferior temporal lobe. The peer-reviewed study selected contained brain tissue samples of both healthy brain tissues and Alzheimer's disease brain tissue. The data extracted from the studies were the methylation status of CpG islands in the promoter regions of the APP gene. The studies were reviewed to identify consistent epigenetic patterns and evaluate the potential role of epigenetic regulation of APP in the pathogenesis of Alzheimer's disease.
During examination of parietal lobe, anterior parietal lobe, and inferior temporal lobe of a postmortem brain tissues of patients with Alzheimer's disease and those without, data in Figure A have shown that CpG islands, a region mostly comprised of cytosines and guanines, are mostly methylated. This methylation was found to be statistically significant only in the inferior temporal lobe. Upon further investigation of the temporal lobe, methylations of CpG at position 60 to 63 was found to have a significant impact on the expression of APP. Additionally, hypomethylations of APP promoters CpGs at the 5' site suggests a reduction in transcription of the APP genes. This would cause an overall decrease of expression of APP in Alzheimer's disease. Coincidentally, another studies have reported that hypermethylation of CpG site in other region of the gene that is not the promoter enhances the transcription of APP genes, which will result in an increase of APP expression. In terms of histone, the histone tail undergoes all sorts of modification, specifically when it undergoes methylation. This histone usually represses transcription. Figure B shows that out of all the histone related to the epigenetic regulations of gene expression, histone 2B residue K108 is the most effective by the methylation process, with more post-translational modification happening in Alzheimer's disease than that of the control.
By examining DNA methylation and histone modification, symptoms of Alzheimer's disease or AD may be reduced before major signs appear. Studies of AD patients revealed abnormal methylation patterns, and hypomethylation led to excess APP protein production, while hypermethylation silenced necessary genes. Histone occultation, typically linked to active gene expression, also plays a key role in AD. This review emphasizes how DNA methylation and histone modification shapes epigenetics. Gene-environment interactions add complexity to disease development, meaning genetic risk doesn't guarantee disease onset. Environmental factors such as stress or poor sleep can influence gene expression via methylation and histone changes. For example, someone with the BIO440 gene might avoid AD with a healthy lifestyle, while another may develop it early due to negative environmental influences. Since genetics alone don't determine disease, diagnosis and treatment must be personalized. Though there is no cure, promising findings show that HGAC inhibitors improved memory and learning in animal models with AD. These results suggest epigenetic changes could be targeted for more effective AD treatments.